Hello, everyone. Hello, and welcome to everyone who already joined the masterclass. And thank you for joining, who's going to watch the recording. My name is Maryna Löwe. I'm a part of the service team. I'm responsible for the project management and the project design for our service projects here at Cube Biotech. And you are here in our scientific masterclass series, which were made by Cube Biotech to enable the researchers from around the world to present their latest results and findings to a worldwide audience, and provides a unique platform for scientists to share their work, connect with others in the field, and receive the feedback from the experts. Before we proceed with today's talk, let's speak a bit about the organization of today's talk. So after the talk, we're going to have a question and answer session. So whenever you have questions, you can use the window in your lower right part of your screen, and then you can switch to the questions and basically type your question to this window, and we're going to highlight this question in the question and answer session. So let's go to our talk today. So we're going to speak today about the next generation antibody discovery enabled by the NativeMP platform and Phage display technology, and an example of P2X4 receptor. So as is the last masterclass this year, so it's a bit more special, let's say so, because we have today not only one speaker, but actually two speakers, because the idea of today's masterclass is to share with you the results of collaborative project between Cube Biotech and ProteoGenix. ProteoGenix is a French-based company which specializes in antibody development, and yes, it's our partner. So with the first part of the talk, I'm gonna tell you the story that's about the P2X4 protein and what kind of the research Cube Biotech did, and then I will hand over to a colleague from the ProteoGenix. So today with us is Conor McCartin. Unfortunately, Matthew could not join us today, but still, I will hand over then, and we're gonna also hear from the ProteoGenix side about the work that they did. Yes, so I'm just gonna start maybe and share the presentation here. So perfect. So before we start, a couple of words about Cube Biotech. So Cube Biotech was founded in 2012, and it headquartered in Germany. So we have our labs and all the production sites here, but we also have a facility in the USA. And here in Germany, a team of 50 people is working to facilitate the membrane protein research. So we're specialized on the membrane proteins, and we already accomplished as a service over 350 different projects with membrane proteins with an impressive 95% success rate. And we also offer products for membrane protein research, and in general also with soluble proteins. We have more than 40 unique products that make the life of researchers way easier. So in Cube Biotech, we offer membrane protein work as a service, so we can ensure successful protein production. So we can go from gene to protein, gene to antigen, or even from gene to structure. So depending on the project, and it's all about mostly membrane proteins here. So first of all, we work on the just a second. Yeah. So first of all, we optimize the expression, and then we proceed with the stabilization and solubilization using our unique NativeMP platform, along with the protein purification. So this is kind of the heart of our services. And of course, depending on the project, we offer different characterization work on these proteins, as well as the structural determination via cryo-EM and X-ray. Besides this, this year we invented our NativeMP target pipeline, where we not only wanted to showcase that NativeMP technology works basically with any single membrane protein, but also give the researchers worldwide access to very challenging membrane proteins that were challenging to work with till now. And yes, so this year we introduced more than 60 different proteins to our portfolio, which are now available as off-the-shelf proteins or off-the-shelf project in case some additional customization is required. NativeMP stabilized proteins can be used in a variety of different downstream applications. So we can go for screening and validation using well-known biophysical methods. We can do structural determination. We can do small molecule screening for the drug development using, for example, ASMS or DEL screening. And we can also do biologics development. So, for example, antibodies or their fragments. So that's going to be the part of our talk today. So, and it works both in vivo as well as in vitro screenings. So let's speak a bit deeply what makes the NativeMP platform so unique. Well, when you're working with a membrane protein, it obviously sits in the biological membrane and you want to isolate it. And the classical way on how you do this, you take detergent to solubilize the protein and get it in the aqueous solution. However, detergent has one pitfall, it washes away the lipid from the protein. And the question arises if the protein is still the same as it was here in the case of the biological membrane. So in Cube Biotech, our philosophy is that we want to keep membrane proteins as native as possible, and that's what NativeMP platform allows us in this case. So because the NativeMP platform, the copolymers, which are base of this platform, they extract the proteins together with their surrounding lipids, and thereby preserve the function, the thermal stability of the protein, and allow you to perform assays even at the body temperature. So how does it work? Synthetic copolymers, they interact predominantly with the lipids in the biological membrane, and they kind of wrap around the membrane protein and form something we call a protein-lipid copolymer complex. And this allows us to work with a full-length and completely wild-type proteins without any thermostable modifications, mutations, or some depletions of the protein parts in order to keep it happy. And the one important thing is here, once you add the copolymers and you solubilize your protein, there is no need to keep some certain polymer concentration in the solution anymore. So basically, once solubilized and stabilized, you can treat your membrane protein as it would be the soluble one. To ensure the success of the stabilization trials, we have here at Cube over 30 different copolymers within 6 different backbones. And in comparison to the classical copolymers that were available a decade ago, they have already way better properties in terms of solubilization. Moreover, that's something also unique for a Cube Biotech. We can offer them also as a functionalized version. So we can, for example, attach a biotin or put some fluorescent markers of if you need to, in case you need to immobilize or you need to detect the protein, or we can also introduce another custom modifications that are required for your downstream application. So let's go to the story of the P2X4 protein. This is a purigenic receptor, and this is one of the best characterized targets here at Cube Biotech. And first of all, what we do with every target basically is after the expression, we take the cell pellets and we proceed with the solubilization screen, because every target or each target is unique, and you need to find the best match between your target and the suitable copolymer. So that's why you proceed with this kind of the screening. So we used here the classic copolymers, and we used here the next generation copolymers, and we directly couple the screen to a small scale affinity purification. So basically what you see here is the elution fractions with the P2X4 protein after kind of first solubilization screen round. And of course, you can see that we have plenty of conditions that can be used further. So you need to be careful. Sometimes the biggest band doesn't mean that the quality of the protein is the best. So that's why usually we take the solution fraction and we try to pre-characterize them in order to narrow down the amount of conditions that could then go for next to step purification, and this would do with the DLS and nano-DSF. So you have some exemplary DLS results here, so which are showing the quality of the particles, so to say, in this moment. So you can see here there is some aggregation. This one is looking better. Yeah. So here we have some signal. Yeah. So that maybe it would tell us, okay, maybe this would be the second choice polymer. And for example, when we see here such a homogeneous particle distribution, that is already a hint for us that we can proceed with a purification. So let's assume from among these conditions we already found something interesting that we want to proceed with. So we take a bit more material in the hand and proceed with a two-step purification. In a particular example of the P2X4 protein, we aim to compare the NativeMP preparations to the detergent preparations. And we saw the differences already in the very first biochemical results. So you see the purification results here. If we take a look on the P2X4 stabilized in our NativeMP system, we see directly that protein is already much purer in comparison to the detergent. But what's also most important, and in the size exclusion chromatography, the peak is very homogeneous when you use synthetic copolymers in comparison to the detergent. So you can see here some shoulder, which probably would need some further investigation, yeah, about the state of the protein in this moment. And yes, and also the stability of the protein. Here you can see that the preparations basically that we see also from our different projects, yeah, so that the preparations in the NativeMP platform are way stable than the preparations in the detergent. So as I said, the P2X4 protein is the most one of the most studied proteins here at Cube Biotech. So we have a lot of application data with this one, and yeah, particularly we'll take a look on how the sample looks like and what you can do with the sample afterwards. So let's take first of all on the results from the DLS and nano-DSF. So what we see here is the two preparations that we did, one in detergent, so in this case, yeah, DDM and CHS, and one preparation in the ultrasolute 18. And what we can see already, yes, in both preparations, we have pretty homogeneous particle distribution. We have a slightly larger particle size for the detergent sample because of the detergent micelle. The interesting part starts when we take a look on the nano-DSF data. So basically the thermal stability, we see that detergent sample is actually starting to aggregate it already around 45 degrees, let's say, whereas the the cumulant radius and turbidity of the solution of the copolymer sample stays pretty much unchanged. What's also very important for the protein is, of course, is the protein still active? And of course, that's something also we take a look at this. So you can see here also the MST, so microscale thermophoresis measurement of copolymer stabilized P2X4 against the antagonist, the 5-PDBD. And what we can see here, that at room temperature, we basically see a binding, and we can fit a binding curve and determine the KD, which was unfortunately not possible with a sample stabilized in detergent. Also, with our other collaboration partner, the ELERIS, we took a look on the ASMS measurements. If just the proteins were in these preparations were fine to use in the ASMS setup, so I will not describe a lot about how it works, yeah, but the idea is actually that when you mix your protein with a small molecule compound, you want to separate afterwards the compounds that were not bound to the protein. So in this case, you go on the size exclusion chromatography, and what is interesting, that actually the recovery of the P2X4 so was way better in the NativeMP stabilized preparation in comparison to the detergent, and also the measurements with the referent ligands, so the KDs that we get were very much comparable to the IC50 values that are available from the literature. And of course, something like cryo-EM is also very interesting to take a look on the proteins. So we have the P2X4 preparations in a different copolymer nanodisk, and from sulfocobipol, we're able to determine the high resolution structure with around 3.5 angstrom resolution, where you nicely can see basically of the glycosylations. We have also a structure with a ligand bound P2X4. So copolymers allow you to go to any of these applications afterwards and, as I said, treat your protein as it would be the soluble one. And of course, not but not least, we sent 2 protein samples. So basically also, again, the NativeMP stabilized and the detergent stabilized P2X4 sample to our collaboration partner ProteoGenix in order to generate antibodies against P2X4. And the result of this collaboration further, you can then hear from my colleague. So thank you very much for your attention at this moment. So I would ask Conor to turn on his microphone and camera, please. Hi, hello.
Hello, nice to join you, and thank you for having me very much. And thank you very much for the wonderful talk yourself.
Thank you very much for coming. So we're going to change the slides, and now I hand over to you.
Great, okay. So thank you again to everyone who has signed up. As was mentioned previously, unfortunately, Matthew, the head of our monoclonal antibody development department, could not be with us here today. So my name is Conor, an account manager within the monoclonal antibody development department here at ProteoGenix. So we are a French CRO located in Strasbourg in the east of France, right on the border with Germany, which has been operating for 20 years now, and which is currently around 200 employees and which is specialized in therapeutic antibody development. So we actually, here at ProteoGenix, we have a fully developed and integrated capacity for every step of the pipeline in therapeutic antibody development. And to give you an overview and provide you a whole range of services from gene synthesis and antigen production to lead discovery via 3 main technologies, which of course include classical hybridoma development, but also more advanced screening methods like phage display, single B-cell screening, and amongst other technologies such as antibody engineering to cover characterization, optimization, affinity maturation, developability improvement, etc., right up to the generation of a stable cell line, enabling our clients to produce the necessary quantities of antibody for preclinical and even clinical studies. Importantly, all of our services are IP-free, so that we can guarantee to succeed, or you do not get paid in order to help our clients to secure their investment. So now we're going to, so here's a little bit of detail on the global monoclonal antibodies market. And I will detail to you a bit more about how we generated antibodies against P2X4 and the rationale that we had behind that. First of all, since antibodies are, of course, a very common and useful tool, I can give some figures on the growing market, commercial importance that they have. The total mAb market is set to reach over 200 billion USD in 2024, with the prediction for 2030 being over 450 billion USD, so more than double compared to now, representing a continual year-on-year increase of around 13%, meaning huge business potential and investment opportunities. Now back a bit more to science. Very important for therapeutic antibodies is the degree of humanness of an antibody. What does the level of humanness mean for an antibody? So here we can see the structure of a full-length IgG with 2 heavy chains and 2 light chains, each made with 1 constant region and 1 variable region responsible for antigen binding. So the first monoclonal antibodies developed in the 1970s were via the hybridoma technique, and thus were fully mouse. However, this, of course, caused immunogenicity problems, as injecting mouse antibodies into a human may, of course, elicit an immune response with associated toxicity and reduced efficacy. The first solution to address this was simply to take the variable domains developed in a mouse and graft them artificially onto a human backbone, giving so-called chimeric antibodies. However, chimeric antibodies still cause immunogenicity problems, and so another improvement on this were humanized antibodies, whereby individual amino acids are changed to make the antibody sequence more human and less immunogenic. But even more recently, we have since developed the capacity to generate directly fully human antibodies for which immunogenicity is not an issue from the go, thus reducing risks and saving considerable amounts of time and money, and to speed up your way to the clinic. We can see these developments in the types of mAbs that are being approved over time. We started out in the 80s with mainly with some rodent antibodies, chimeric and then humanized antibodies, with fully human antibodies now taking more and more space in the increasing amount of mAb approvals, and which may become more dominant within the next decade. So the question thus remains, how do we develop these fully human antibodies, especially as we cannot, of course, ethically directly immunize living humans and then isolate them? The solution to this is, of course, phage display. So basically, phage display is a wonderful technique which won the Nobel Prize in Chemistry in 2018, whereby we can take a bacteriophage, a virus which infects bacteria, and so which is of no danger to humans, and modify the wild-type P3 protein of the phage to form a fusion protein with an antigen-binding antibody fragment. Since there is no species limitation, we can modify these phages with entirely human antibody fragments, which we can use for the development of fully human mAbs. In order to construct a phage display library, we isolate B cells from human donors, extract the total RNA to then synthesize cDNA, then PCR out the variable chain sequences responsible for antigen recognition, and clone these into a phage vector encoding the viral phage proteins in order to create a library of phages displaying antibody fragments corresponding to the immune repertoire of the isolated human B cells, which can then be transformed into a special bacterial strain for use. One example of such a library is ProteoGenix's high-diversity naive human library, which has been constructed from around 500 healthy donors from 5 different ethnic backgrounds in order to maximize the diversity of the library, as there can be significant variability in the immune repertoire of individual humans. This has allowed us to obtain an exceptional library with over 60 billion distinct antibodies, providing a great source of therapeutic antibodies with no humanization necessary, thus boosting your way to the clinic. So how then do we use this library to get out fully human antibodies? This process is known as panning, whereby we first coat our target, which may be a protein, a peptide, or a small molecule, to which we then add our phage library. The phages which carry antibodies specific to our antigen will bind, while the majority of the phages, which are non-specific, will be washed away, allowing us to identify binders of interest amongst billions of different antibodies. The bound enriched phages against the target can then be eluted at acidic pH, then used to reinfect bacteria for amplification and restarting of the cycle. This is repeated for several rounds to continually enrich the phage library against the target of interest. Following the conclusion of the panning rounds, a limiting dilution of the enriched phages is carried out to identify monoclonal phage binders corresponding to one sequence, which are then screened by ELISA and sequenced to get the final specific antibody sequence. This is a fantastic method of quickly, within 2 to 3 weeks, getting a selection of fully human antibody sequences against a target of interest. In addition to this panning process, we also have solutions for difficult to express therapeutic targets, typically such as multipass membrane proteins like GPCRs, which are major targets but cannot be expressed freely in soluble form by classical recombinant protein expression. Thus, we have a range of solutions that we can use to get past this problem, but in order to identify binders against these targets. However, they all have limitations. i.e., if we use micelles containing the target, these are generally very impure and unstable. If we use short peptides corresponding to a part of the extracellular domain, they're easy to produce, but conformational bias due to their linearity means that they may not produce relevant antibodies against the actual native protein. And then we can cells overexpressing the target. And this indeed will give the antigen conformation that we require, but it is very expensive, requiring stable cell lines as transient expression is generally not enough, and it is difficult to use as the use of a cell in a panning process, of course, generates lots of background, making it difficult to identify relevant binders. And that is where Cube Biotech's nanodiscs come to save us, as those represent the perfect fit as a solution for all of the aforementioned problems of the other different technologies. Nanodiscs are very pure and stable structures, They give a fully native target structure and function of the protein of interest. They're easy to produce, in particular in comparison to stable cell lines, and they are very easy to use with very limited background generated during the panning process. So we decided to collaborate with Cube Biotech on this, and we decided to use human P2X4 as a proof of concept for our use of nanodiscs in our Phage display projects, which, as Maryna said, is a multipass protein of major interest in oncology. So Maryna's team have produced a human full-length P2X4 embedded in a nanodisc with a C-terminal Rho tag for coating into a ELISA plate via an anti-Rho antibody. This enables correct coating and orientation of the nanodisc on the plates in order to drive selection of antibodies against specifically the extracellular region of interest. As without this specific Rho-orientated coating, we would get antibodies against both sides of the antibody, which would not be of interest for the project. For this, and for these particular type of panelings in general, we have adapted a specific Phage display protocol, whereby we first carry out a depletion step of our Phage library by incubating it into a plate coated with the control nanodisc containing a completely unrelated protein, and then pipetting out the cleaned library, which would be depleted from binders against the blank nanodisc. We also, in the panning process, carry out depletions against the anti-Rho antibody, which is used for the nanodisc coating, as during the process we may also naturally get enrichment against this protein which is present. The whole process which I described before is then applied, which means incubation on plates coated with P2X4 nanodisc, wash of unbound unspecific phages, elution and repetition of the whole cycle, and then single phage clone screening by ELISA followed by sequencing, which allowed us to identify successfully 2 binders with strong binding against P2X4 nanodiscs and no binding against the control nanodisc as desired, making it a very successful project for us indeed. Once these 2 binders were identified and sequenced, we expressed them as full-length recombinant human IgGs by using our proprietary extensio system optimized for transient expression with high yield, high purity, and low endotoxin, with the 2 purified antibodies then being tested via ELISA in order to confirm that we had the binding of interest. Here you can see these ELISA results showing high affinity binding of both antibodies at a concentration of 1 microgram per ml and below, with high specificity as no binding was found against the control nanodisc containing the unrelated protein. So these results provided proof of concept that fully human, high-affinity antibodies can be obtained against complex membrane proteins using a combination of the nanodisc platform of Cube Biotech and the Phage display platform of ProteoGenix. Very importantly, the two anti-PTX4 antibodies we obtained are free of IP and available for purchase. So if you have any interest in them, please contact me using my email address displayed here. As a reminder, these 2 antibodies are fully human, so no humanization is needed, and were developed using native full-length human PTX4, and so many risks have already been removed. In addition, P2X4 is a hot target with very limited existing antibodies, of which most are poor performers due to the use of irrelevant antigens like peptides. So for sure, the antibodies we have identified have high therapeutic potential. And that is all. Thank you very much for your time.
The microphone, important. Yeah, thank you very much also for your talk. It's exciting to see that it works so perfectly with the nanodisc, basically. Yes, so we have some questions already. I see they're popping on the screen. So I would ask the team to showcase the questions. So the first question: hello, can you use any types of antigens with your library? So I think it's more to you, the question.
Indeed. Essentially, yes. So we have successfully used our libraries against recombinant proteins, against small molecules, peptides, nanodiscs. So the answer is yes. We can essentially pan against anything, glycans as well. I do believe. Some are more difficult than others. So of course, a large recombinant protein would be a bit easier than a small molecule, but we have protocols in place for these to get out binders, and we're always up to the challenge.
Okay, thank you. Next question. Hello, could you explain the difference between Cube's nanodisk and MSP nanodiscs? Great question. Thank you very much. So we prefer not to call the NativeMP stabilized protein as a nanodisk, because there is exactly this problem comes that people know actually the MSP nanodisk. So there is sometimes we need to clarify about what kind of a nanodisk we're speaking at this moment. So MSP nanodiscs are generally from protein origin. So they are coming from the apolipoprotein, and they form a kind of also a belt. So it looks pretty much the same as a synthetic copolymer, so which wraps around, but it's from proteinaceous nature. And the preparation is very, very different. So this is the most crucial story about the difference between the NativeMP, synthetic copolymers, and the MSP nanodisk. So in order to proceed with the MSP nanodisk reconstitution, you need first to solubilize your protein with a detergent and purify it. And then you need to make a screen for MSP nanodiscs. So that means that you need to find a right size of the disc, and the right lipid composition inside, which is different by NativeMP platform, because the synthetic copolymers, they basically adapt to the size of the protein, and the discs that are formed, they are then protein specific. So that's why we actually measure the DLS. So the both preparation can be seen as nanodisk, MSP and NativeMP platform, but the nature and the preparation, is very, very different. So it's important not to, it's important to say what kind of nanodisk you're using in this moment, And also a big pitfall of the MSP nanodisk is the moment that you actually, while doing the reconstitution from the detergent to the MSP nanodisk, so you have a very, very severe loss of the protein in the end. So you need to purify more in order to get some substantial portion integrated in the MSP nanodisk, which is not the case for the NativeMP platform. It works in a single step and gives you nice stabilized particles with a protein that never saw detergent before. Okay, how many rounds did you perform, and how many clones did you test to eventually detect 2 binders?
So I did not directly manage that project myself. However, that's definitely information that we have on hand, and we can get back to you with that information by email. That would be no problem. However, we generally perform 4 to 6 rounds of panning for projects. So we follow the polyclonal enrichment as we go along, and once we decide that it's enriched enough that we have a variety of binders without totally killing the diversity, then we decide to stop. We rarely go beyond 6 rounds, essentially.
Yeah, thank you. What is desired therapeutic indications for targeting P2X4? Yeah, well, P2X4 is neuropathic pain receptor, and it's also associated with several cancer types. So as a prognostic marker, so it's in general a kind of an interesting target to take a look at. So in the end, so I hope this answers the question. Hello, do you have only one or several human libraries?
So we have several. So we have our main high diversity and naive human library, essentially, but we also have other libraries which are targeted towards specific pathologies. So we have our cancer patient library, which we have constructed from PBMCs of cancer patients with a variety of different cancers. And so with the idea being that the library is enriched against specific cancer neoantigens, which are, of course, very interesting therapeutic human targets. We also have a human autoimmune library, which is taken from patients suffering from more general breach in immune self-tolerance, and thus likely having a greater repertoire of anti-human antibodies against a wider variety of targets. And especially particularly in cases such as pandemics. I know that we, very early on in the pandemic, we constructed a COVID-19 library from COVID patients, and we did we did a fair number of projects with that library during the height of the pandemic to identify anti-spike protein binders and things like that. So and I hope it won't be the case, should we have another pandemic like that, we could quickly construct a library to help with that.
Oh, wow, that's interesting. Yeah, so that's... How much antigen in nanodisk is needed? How much non-specific black nanos are needed in your method? So I think, Conor, it's also the question to you.
So around 500 micrograms to 1 milligram would be around what we're looking for a panning program. And the specific blank nanodisk around roughly the same. Yeah.
Okay, thank you. How much can you control the size of the synthetic nanodisk, which you can control with MSP? Concerned about monodispersity. A great question. Thank you very much. So in general, the size of the particle, so of this copolymer lipid protein particle, in the end is very copolymer and target specific. So that's again the reason why we measure DLS. So we pre-assist a kind of a first quality control of the particles that we see, and usually almost in all our preparation we see a very homogeneous samples in terms of the size distribution. Yeah? You can theoretically control the size, so that's something that you need to explore during the solubilization screen, for example. So you can play around with different concentrations or the temperatures, yeah, different conditions that you test in this moment, yeah, to see if you can change the size of the disk. Yeah, so it's possible. It's, of course, you will not get the disk like that's gonna be like, I don't know, 3 times larger, yeah, so, and then 1 times larger, smaller, yeah, so in the end, but you can make a space in the nanodisk itself, yeah, so to let your protein move a bit. So some proteins would require, for example, certain conformational changes and the movements in the membrane. So you would probably search for the particle distribution that would be probably, yeah, in the hydrodynamic radius a bit larger, yeah, and then purify those and characterize it further, yeah, so to see if this is a suitable preparation for your downstream application. Yes. Did you fuse Fab or full-length IgG into P3 in your Phage expression system? I'm sorry, maybe I read it wrong now, but
no, that's absolutely a question for me. So neither. We use an SCFV. We also have Fab libraries available, but we have a general preference for SCFV as we find we get better enrichment. As far as I know, full-length IgG Phage libraries are not used. They might exist theoretically in the literature, but they aren't used in panning projects, as the Phage it's a very large particle. It's 150 kilodaltons. The Phage is the virus doesn't like producing it, essentially. So you kind of need to go with a smaller format, and then afterwards, once you sequence the phages and you want to express the antibody as the full antibody, you graft it onto a backbone with the rest of the sequence, and this works quite well.
Thank you. Next question. With or without interchain disulfide bond in your Fab library design? That's also, I think, the question for you.
Which does not specifically apply because it's not a Fab library indeed. So there is no, as far as I know, there's no interchain disulfide bond for an SCFE. There's a linker. So with the SCFE, you have the VH and the VL domains, which are joined by the addition of an artificial linker. So it's expressed as one molecule.
Okay, thank you very much. Do we have more questions? Can I get the recorded meeting at the end, so by email? I think not only this, so you will, I think, have the link, yeah, so for the recorded webinar, but also we have online stream right now on LinkedIn, and the recording of the masterclass is also available on LinkedIn on the Cube Biotech site. So you're welcome to follow Cube Biotech and, of course, ProteoGenix for today's webinar, and of course you can watch the webinar there as well. So I think that's all with the questions. So I want to thank Conor. Thank you again for coming on such a short notice. Yeah, that's
thank you very much. No problem.
Yes, it was very nice to see you. I hope on the productive collaboration in the future between our companies. Of course, if you have any questions left that you want to ask address to Cube Biotech or ProteoGenix, you're free to send us an email, or you're free to comment the recording of our webinar, and ask your questions. So we're happy then to answer them either in LinkedIn or per email. And in the end of our webinar, I want to also make an advertisement for the next webinar. Can we show up this one? Okay, that's not available. I'm sorry, but okay. We will have next webinar in January next year. So stay tuned. Take a look on the Cube Biotech website, so you will see the advertisement directly on the homepage in the upper corner. And I thank you very much for being here today. I wish you great holidays, Merry Christmas, and a Happy New Year. And we'll see us next year again. Bye-bye.